You guys might want to move in here so you can see this a little clearer. It's probably going to work better if we dim the lights as well. I'm going to talk a little bit about uh, some of this. Originally, they contacted me to talk about composting, and that's a week long, if not a month long, if maybe a lifetime long learning. But what I have learned is no matter how you choose to compost or what you do, and no matter what materials are put in there, a successful compost should kind of have the same result in the end. You're going to have a healthy microbiome that your soil is going to like, and that should be in our soils anyway. In a lot of cases, you just don't see the full soil food web, and nothing but, very, very healthy soil. And a lot of that is just in our management. So really what we're doing with a microscope, it's just another tool. And just like the Hubble telescope looks out and they just keep finding more and more and more, this is the opposite for low knowledge. It's even more and more and more. And it blows your mind. We don't know much about the soil microbiome. I mean, we'll see, they talk about billies of different species. We'll see, or not see, but the numbers in a teaspoon of soil, and it's actually true. So hopefully today the point is just to show you that they're there, they exist, what some of them look like, what should you be looking for. And I want to spend a lot of time just looking through the microscope, and I'll try to, point out what I see anyway. And I got my training on this kind of during COVID. I took the soil food web class from Dr. Lane. Anybody in here take that? You took it? It's excellent. It's college level training, if not higher. It's good to have a background in the soil health principles, which I felt I did. So that was really good information to enter into it. But then what are we really trying to accomplish? What's going on in the soil at that lower level? And I better get started here. We'll never get through it. Okay, advance. Those failures of water. Yeah, too late. There we go. So I've stolen some of these images from Matt Powers. I also took his microscope class. And for any of you that are interested, these are the best resource I found about composting and methods to get a regenerative soil. And that picture's right in here. He shared them with me and said I could use them. But he does a great job of explaining all this information in one place. How does compost, how do nutrients, how do all that play together into making a regenerative soil? And, it's not just the microbes, it's all of it together. And he also wrote a book on microscope, full of information. I mean, way more than you can, this would be for someone that has a microscope and wanted to have a little bit of a resource as to what am I looking at, and also some new ways to evaluate, or a new approach. The soil food web class teaches you how to look at something and count it, and Matt is more of let's observe and recognize that it changes. And that resonated with me a little bit better, because you look at soil, you just get your frame of reference. What am I looking at? Where did it come from? What time of year is it? What should I be seeing? And he does a really good job of helping me go through that. It's more of a observational method than it is a counting, because you can look at something and 20 minutes later it'll be something completely different when it comes to these microbes. It's just fascinating. So he compares, the soil, or your sand, silt, clay. We've all learned that in college. That's what the soil is. That's what you got. That's kind of like take all that work. And then the soil food web is the actual operators that put it all together and work with what they have
They, so we're managing that community for a better outcome in the end. I just thought that was really interesting. And it is kind of interesting. The computers work on silicate, and so do the microbes. And there's the soil food web. We probably have all learned about this well, maybe not every, NRCS teaches the soil food web, and it all kind of starts with bacteria up there. You see a lot of arrows pointing to bacteria, and we'll look at some bacteria. Bacteria form a relationship with the plant roots. The plant roots feed the bacteria, root exudates, and it starts this whole food chain going on down. And as you go, less and less nitrogen is needed for the higher levels of life. And as a result, they poop it out. What do you do with excess? You get rid of it. And that's the soil food web. It's the basis for all of the organic matter, all of the humic substances, all of the aggregation, all plays into that fully functional food web. And a lot of times when I look at soil, we're getting cut off somewhere up there between the fungi and the protozoa. If you drew a line, that's what I'm seeing in a lot of soil. We're missing the rest of it. Little bit more on that exchange at the plant roots. And we'll look at this, hopefully, if I get moving along. But this is kind of what's happening right in there with the right at what they call the rhizosphere, right around the roots. And it starts this whole food chain, the rhizophagy cycle. And this is really getting right down into the very tip of the root hairs. That's where the root exudates are coming out, feeding this bacterial population. If you've never heard of this cycle, check it out. This is how it works. And you can throw everything you've learned at the university about plant nutrition right out the window when you recognize that this is how it works. If you get this working, the plant actually farms the bacteria, takes them in, dissolves the cell wall, spits out whatever it didn't need. It's taken the nutrients from that bacteria, and it populates again. It starts this virtuous cycle, so to speak, that the plant gets what it needs and the microbes bring it, and the whole system is working there nutritionally. It's not to say that you don't need nutrients. You still need to have something to work with. But the microbes can do that, and the plant knows what it needs better than we do. So I'm really kind of stressing this whole thing because this is relatively new science. It's 10 years old. So we're going to have to rewrite every textbook there is and take this into account once we figure out what it looks like. And we'll look at it under the microscope. I'm looking here at it, you can't see it. Right here. That's a plant root, the picture that I took of the root hairs. They're just wet, glistening with this glucose type. I mean, there's a lot more than just sugar in there, but the plant specifically farming the microbe community that it wants, and the community forming, and then the food web starting, and you get this soil aggregation going on. It's pretty fascinating. So we want to see a lot of bacterial diversity. This is a picture I just took, and we'll see some of this live. Everything in here is bacteria, and lots of different shapes and sizes. And if you want to count them, go ahead. That's not for me. What you would have to do in the soil food web is dilute it down, figure out an area, count how many, and then you do the math. And they've got tools that help you do that. So you can determine kind of whether you have more than you had before. But 20 minutes later, like I said, that number changes. So, but and you can start seeing it clumping together. That's really just the beginning. And the golden color is also something that we're paying attention to. Beneficial fungi is something we want to see in the soil, and it's pretty well indicated by something. This is a septa up here, kind of a like a dam in the flow, and it can control things in and out. You see the U-shaped one. Those are beneficial fungi. They generally have some color. Not what I thought we were looking for when we're looking for fungi, but that's what you look for in the soil to indicate that they're there. And then the dark, really dark is well, I'm not sure about that one. We'll see some stuff that looks like that, but I was going to say it's a fungal spore. But a little further up that food web, this is a testate amoeba. And you like to see 2 or 3 per view. That's a pretty good population. It indicates that there's oxygen and air exchange, and they're there, they're happy, and they're doing what they do. They eat a lot of bacteria and start that poop loop, as they call it. Flagellates and ciliates. Now we're a little further up the food chain. This is actually a stalked ciliate. If you can see he's connected to a chunk of organic matter. They're a little bigger. Again, they eat a lot of bacteria. And the ciliates are not real distinct, but like these little dots. And in a good, lively compost, you'll see them kind of moving around slowly. They don't really know where they're going. It's because they only have one little whip to move around. Whereas a ciliate, when they're moving around, they have hair all the way around that they use for motion. So they kind of act like they kind of know where they're going. And then the next level is the nematodes. And there's a lot of different kinds of nematodes. Spent weeks learning how to identify nematodes, good from bad. Or I came to determine that there really isn't good and bad. It's just what you see as a result of the condition. And the nematodes, even, this was new that I learned from the Matt Powers course. He said they can change function. They can be, they adjust. They can be eating bacteria one minute, and then conditions change, pH or something, it rained, whatever, and they'll go and start feeding on fungi. So there's bacterial feeding fungi. There's fungal feeding fungi, or nematodes. I'm sorry, getting my bacterial feeding nematodes generally are considered very, normal. You want to see some of those. The fungal, they're fine. It means you probably have a lot of fungi, which is a good thing because they're there to feed. And then you have the root feeders, and then some predatory ones as well, where they'll eat about anything. The root feeders are the ones that can cause you some problems if they get out of hand, but still, a few in there I think are probably normal for a healthy system because everything has a job to do. Little for higher up the chain, and then there's be the things you start to be able to see with the naked eye, really. Rotifers, microarthropods, and earthworms. This is a microarthropod. You're looking at his underside. You can see the legs. Bigger yet. And this microscope has some neat features, and one of them is this unit here. And it allows you to put phosphorescent lighting so you can see just a different glow to things. As it's just really a neat picture, but really what stands out, just like black light, phosphorus will glow. So you can check and look at your corn root to see if it was inoculated with the fungi. So that's a picture of what that looks like, and hopefully we'll be able to see some of that too live. But with that light, you can determine whether your inoculant did any good. If you put some microbial inoculant on your seed, you want that happening right at germination, and you can actually check. The other thing you can do is just look and see, is it starting to build soil aggregates on the root. And this is just another option that you can do as a top light. Not everything has to come through the bottom light. This is a top light. And you really get some interesting pictures with that. So we'll look at that too. This is actually wheat straw in the fall after it's been applied with a fungal rich decomposing. You've heard these products on the market now that decompose residue. Yeah, that's real. There they are, doing the job. That'll be gone by spring. So now I have to switch to the microscope. What do you want to look at? I've got just some tools of the trade over there. It's different things. You can check it out, a pH meter and a salinity meter, and all ways to just evaluate how's things looking in the soil. And I found that you can get these little garden test kits too that you can test nutrients, and they're awesome. I mean, for 20 minutes you can figure out NPK for whatever you're working on. I use it for compost to test the quality of my compost. And your compost isn't all created equal as far as pH. And sometimes we're moving things the wrong direction with the compost we're using. So you want to check the pH of your compost. Okay. I think we'll see if we can spot some nematodes. This is a collection from it's just out of a filter where I'm keeping an aquarium. I've got some fish alive, and that's really a good place to look for the microbial community. Even though it's in the water and it's generally anaerobic, there's an aerator system in there, and I've just noticed that there's a lot of nematodes in there, and they look like they're bacterial feeding primarily, so it's pretty rich in bacteria. Okay, we're at right now this is a 10x view, 10 times, and we're seeing some things. Those larger swimmers, those are rotifers is what they're called, and eating bacteria like crazy. There's a big one up there. Let's see if I can get this to work. That would be helpful. Oh yeah. So the other thing this microscope has is this 4K camera. That's kind of the expensive deal. And I can snap a picture of anything and it goes onto a USB drive and send it to somebody that knows what they're looking at and get answers. It's just so handy. But this mouse is a little wild. It's backwards, actually. This might help me. Now the mouse goes where I've taken it. You see that one there? He's just starting to get going. Not probably the best thing to spray on plant leaves, because this is when you see a lot of bigger things moving around, that's moving anaerobic, and that community really isn't beneficial on your plant leaf. So you really probably wouldn't want to spray this on your tomatoes. But into the soil, it'd be fine. It'd be fine in the soil or in the off season or whatever. It's just okay. I'm going to go back and show you how I would scan this for nematodes. You find a corner. I had it right the first time. Find a corner of the cover slip, and they call it mowing the lawn. And you just kind of go through there, and I'm looking for movement, but of course nematodes, they're long and they're generally pretty good size. So it turns out it's the hairs on warehouse beetles. They're they're like maggots and they have hundreds of hairs. And that's what those are, is the hairs. And it's in the fish food that I was feeding. So it's a processed fish food that's pellets that's made out of those beetles, apparently.
What kind of fish do you have?
Goldfish. They're just and they love them. I mean, they'll eat them, but they're not getting them digested. Those hairs they're going through the fish gut. Just interesting. So that's enough about goldfish. I want to look at, I have 3 different soil samples up there, or compost samples would be a better description. I better put the lid back on this before I spill beetle setae all over the floor. How am I doing on time? I got 3 good ones, and you guys can decide what you want to look at. This is a Johnson Sioux compost that's about a year old. This is a worm bin collection I took just yesterday out of my worm bin. There's worms in the bottom of that one. And this is a commercial product that's very, very highly mentioned when you're searching good compost. This is one that they're doing a really nice job. It's called Catalyst Bioamendments, one of the better companies. But
Sure, you want to see a new one and an old.
A new one and an old one.
Your current and sometimes your.
So the Johnson Sioux is the oldest, and the worm bin is yesterday.
You did the Johnson Sioux one?
Yeah.
How'd you do that? Did you keep it inside or in a garage? Or
Oh, this is what I made all last summer.
Okay, so it didn't go through overwinter. You made it fast enough. It got done before it froze.
Well, yeah, this one actually, it was about 9 months. I felt it was complete. I mean, it continues to evolve and get better. The longer you let it go, the more soil-like it becomes, but the less biology you have. So it's just constantly evolving. That's the way it works. So the way what I generally do, I don't get too carried away with measuring exactly, but I've done it enough where I kind of know what's going to be a decent dilution. I'm sorry, putting it in the wrong deal. Just a test tube, and I'll take about 1 gram and a little more, maybe. That's too much. If you get it too much, you can't see anything. It's just so busy that you can't see anything. And my water, I have a bottle over here. About a 10 to 1. 10 to 1 soil is what I'm looking for, and water. And this is I should open this. It's you can use tap water, but I mean, some municipal water has a lot more chlorine than others, and you can just treat it with a little bit of humic acid. That takes care of the chlorine if you are worried about it, but tap water works pretty good. And then the way they teach in the Soil Food Web School is just a gentle 30-second back and forth. We don't have time for that.
And I would hope to see some gungle in there. Oh, there's a chunk right there. Really nice one. You can see that the pieces are a little bit more organized. The clecks darker.
And that's your soil building process. That's how soils are built. You have a parent material, yes, there is geology involved, but this is soil building. This is the humus. It's at the top, and it's building from the top down. This starts going deeper in the soil. So that's what the regenerative process that we're after. Yeah, this is a terrible sample. I'm sorry about that, but there's a fungal piece right there. I'm going to get over to this side. Maybe it focuses a little better. Good testate amoeba right there in the middle. Good to see. means oxygen. There's another one. A lot of bacteria in there yet. A lot of diversity, I would say. I mean, it's probably on the bacterial side yet because of its young age. There's a tool over there called a microbiometer. Has anyone heard of that? It's a way you can quickly gauge your fungal to bacteria ratio. You put a little, I think it's salt basically, into your test tube, and you agitate it for a certain amount of time, and then you put a drop on a test sheet and take a picture with the cell phone, and then it'll instantly give you a readout of your fungal to bacteria ratio. It's pretty good. The only problem I have with it is it doesn't really tell you what fungal community you're looking at. So you could put a sample in there that's full of Phytophthora, and it would be a high fungal. Oh, I got great fungi. No, you got a problem. So this is a fungal there. This coming off of here, and another piece there. There's more fungi in here than this. Actually measured 0.6 to 1 when I ran it through the microbiometer. This sample 0.6 to 1, which is pretty darn good for a compost. It should get better with more age. And every time you stir it, you kind of mess things up too. So more of these testate amoeba, 2 different styled ones.
Think there are a lot of different like crop rotations and managements. And have you seen
A lot more fungi in the
Yes. I mean, the most fungal you'll see is in a forest. And, it's fungal for a reason, and you probably couldn't grow very good crops there. It's too fungal. And so the Elaine Ingham class teaches you're looking for about a 1-to-1 in an annual cropping situation. Now, I can tell that what we're doing is working when you have that. But, can I get that on a treated seed? And you can, but you need to be able to prove it and look at it. And so the first thing here is we'll do the other light source. And this costs about $8, this light. So, and it's one of the coolest pictures you'll ever see, really. All those root hairs, all that biology forming right on the root hairs where the exudates are coming out. There's the bare root in the background, and we're looking through depth here, focusing. I'm very curious how this is going to look under the other light right here. Turn him off. Turn him on. Why won't he come on? Uh-oh. Follow my cord. That was working last night. Wonder if this doesn't have. Should be working. I hope my bulb didn't burn out. That's not good. Now it's working. Oh, I know why. I'm too far out. Never mind. Well, no, I'm losing it entirely. Like there's no power there. Huh. One I'm going to have to figure out. Anyway, you can kind of see what's going on here. You see the darkness around the individual cells, and under the phosphorescent light, that all those little channels would be like in here. All of those little channels, they kind of call them train tracks. It would all be lit up and glowing, like I showed you in the other earlier picture on the PowerPoint. And that would mean you've got 100% inoculation in that root when you can see that. Sometimes you see partial, especially treated seed. So there's a little bit of a battle going on, I think, on treated seed, for fungal, but it can still happen. And really, what overcomes it all is healthy soil, and because they got everything they need to make it work, or healthy compost. I do not know why something got bumped or what, but I'm not getting my light. I'll figure it out later. Back to that top lit. Oh, I know why. It was on. It's on. Right there. No, it isn't. I don't know. Generally, things don't grow yellow like that unless that light is on, but it sure is. It's really busy going on there at that rhizophagy. And it just really makes it real when you can see a picture like that and see all of that, all those networks being formed, and the communication back and forth between the plant and the microbe, and then the whole community of life that is supported by that, all to the benefit of everybody. There's another piece. I'll see if I can find something on here. On these roots. Yeah. I was trying to find a root a little closer to the seed. When the seed first germinates, I did it just in a wet napkin, really, and you can see all those hairs. I mean, it's just wild. And now they're all completely covered, closer up to the seed. So this is right out of where the seed started sending roots out. Let's see if we can get a good lighting on that. And if you actually took this and put it in the test tube and shook it up and knocked all the microbes out of there, you'll end up seeing that same community of microbes that you want to see. Yeah, well, not this one. I had a different microscope to take Elaine's class, and then I started taking the Matt Powers class about 2 years ago, and he's the one that convinced me that this is the way to go, because it's probably not anything you really need, but I'm curious, and I couldn't get enough of it. I mean, and then to be able to communicate this a little bit and have the chance, it's just, I feel good about it. I don't think I wasted my money, but how do you turn it into an educational piece that it's just a tool, but I think it makes things a little bit more real. We talk about that microbial community in the soil food web, and it's just like, yeah, whatever. But it's real. And our management affects them. There's no doubt about it. So it's rotations, it's back to the soil health principles. And then we have lots of tools to help restore and support that community. I know you hear bugs in a jug. We have a jug, but we call it culture or creatures in a culture. So this is our microbial inoculant it's about, this is Elevate Ag I'm speaking about, but we can take this and feed it and expand the community right in the soil trench. You don't want to put the two together, you can't, or this jug will blow up because it'll expand so fast. But if we can put all that in a sprayer and get it in the ground, and if we have time, I've got one that I prepared. This is right out of this jug, and it's really not all that impressive. 2 minutes? Okay, I'll hurry. It's the whole point, right? If I can't sell you something. No, I'm not here to sell something. I think it's interesting to observe it and see for yourself.
And I think those things like that can accelerate it. It just has this degraded soil. I mean, it can help build the right environment. It's kind of like, if you build it,
Yes. But we got to quit doing the bad things too. So you can't, and I think that's just where we are in so many no-till systems. We're doing a lot of really good things, but we're doing things that just kind of keep you stuck because we're cutting off that larger community.
Yeah, I had a producer that had livestock integration, cover crops. He had 3 crop rotation. Alfalfa was periodically added. I mean, like, that's pretty diverse around here. And his actually was one of the lower fungal ones and like a symbol for a bean, I mean, it wasn't just that point in time. I mean, I only did it one time, I mean, it's so, anyway, it just, like, it
mean you work feats and it did help me figure it out, but maybe realize I don't know what's going on.
Wish I'd had my microscope so I could have looked at soils.
So this is our microbial concentrate. This goes out at a half gallon to the acre in the furrow. I mean, okay, and the guy that formulates all this for Elevate Ag has figured out how to put things to sleep. There's nothing going on here. Everything's asleep. And that's proprietary. But he said there's 4 ways you can do it, and he told us 2. One is sugar. You can overwhelm with sugar, or you can change the pH enough to where nothing's alive. So what we do when we want to feed this now is we add the foods and we add water. Okay, this is a fed community now here. You start seeing the movement and you start seeing, I mean, look at how many microbes are in that 400X. I'm shaking, but, okay, I'm going up and down through that drop. And the diversity that we were talking about earlier.
Little kinds of cruisies.
Yeah, they move around, those bigger bacteria. Okay, and then back again to the ones that are asleep. I never went to the higher magnification and thing. I mean, there's some vibration to it. If I can get focused in here. You see things lick up there at the top. They're not totally stationary, but they're certainly quiet. And when we feed them and give them water, 2 things that are changing their condition, the population goes crazy. And you want that happening right in the furrow, right with the seed to support that rhizophagy. And these microbes are actually originating out of a compost. Put the lid on, Jim. And it's a compost derived, so we don't know what's all in there. It's just a big, diverse community. We had a DNA run on it, and they can't, 20,000 species, most of them we don't know what they are. They don't have names. But that's that diversity. And then they're fed kelp and some sugar, molasses, and some fish hydrolysate in the compost pile. Then they're put to sleep so we can ship them around the country. And then we have another product of foods. Guess what's in the foods? Kelp, fish hydrolysate, and what was my third? Sugar, molasses. So the microbes, if you just throw them out there, where's my kelp? Where's my, and different microbes want different foods. We don't have all that figured out, but the complexity of the diversity of all that. So they're trained to eat that. We give them what they want. And then in the meantime, the community is adjusting to where am I, what's here, and what can we do? And what's this plant telling me that he needs that I can help bring? It's just fascinating. So the whole thing is recognizing that there's life, celebrate life, and that it's a different approach to agriculture. So that's my talk. Thank you.